Hello and welcome back. In this video, I'm going to talk about the male and female reproductive systems. I'll probably divide this video up into two parts, the male reproductive system and the female reproductive system. This particular system is one that's interesting to teach because a lot of people think they know everything about the male and female reproductive system, but it's a very complex system. There's lots of parts to it. I'll just give you an introductory view of these systems and let you enjoy some of the complexity of them. So the male and female reproductive systems are a series of glands and tubes that produce and nurture sex cells and transport them to the site of fertilization. These tubes and glands, though, are very complex. They have to be controlled, highly controlled and organized and timed with each other. So the male reproductive system is what I'm going to focus on mainly in this particular part of this lecture. And the jobs of the male reproductive system aren't as complex as the females, although I don't want to disparage that system because it's still super complex. But the male reproductive system's job is to produce and sustain functional sperm cells. Then, not only do they have to produce and sustain the cells, they have to transport them to the site of where they can fertilize an egg, so outside the body into the female vaginal canal. So there's a lot of parts to the male reproductive system, and what I'll do is kind of give you an overview right here and then go through and talk about each of the parts more specifically and some of their jobs that they perform. So I guess you could start anywhere looking at the male reproductive system. So covering over the parts that are producing sperm cells is the scrotum. The scrotum is a flap of skin. There's some muscle tissue associated with it, and it protects and covers over. If you notice, the scrotum is going to allow the testes to hang outside the body. They're not actually inside the cavity, but they dangle outside, and we'll talk about the reason why in a little bit. So the testis is singular, testes is plural. The job of the testis is to produce testosterone and to produce sperm cells. The epididymis is going to be the part that the sperm cells move to next, and this is where they will mature and be stored until ejaculation occurs and the movement of the sperm cells when the sperm cells move to the outside of the body. We then have the vas deferens, also known as the ductus deferens, and this is a tube that carries sperm cells from the epididymis eventually to the ejaculatory duct and then to the urethra to be taken out of the body. So if we back up just a little bit, we'll see that the seminal vesicle or seminal gland, it produces a fluid that helps us to sustain and nourish the sperm cells, and it will be added to the sperm cells as the sperm cells come down through the vas deferens and meet the seminal fluid, vesicle fluids in the ejaculatory duct. We also have the prostate gland. This is going to also add fluids to the mixture that we call semen. And one last little part before we talk about the penis is the bulbourethral gland. This is a gland that adds mucus, so it kind of lubricates the urethra with mucus. Let's see if I can change my pen here. So the bulbourethral gland will actually lubricate the let's see if I can get my pencil right here, get my highlighter. It's not letting me do white, so let me go to my pen and see if I can do white here. So the bulbourethral gland will add mucus so that the sperm cells can flow very easily through the urethra, but it also adds mucus to the tip of the penis so that during sexual intercourse there will be less friction generated. So if we get down to the last parts, we will get down to the penis. The penis is the cylindrical organ that kind of hangs outside the body. Of course, it fills up with blood and becomes erect, so it can become functional to transmit the sperm cells to the vaginal canal. There is erectile tissue. The corpus cavernosum and the corpus sponginosum are erectile tissues. They're essentially cavities that fill with blood so that the penis can become erect. Without the blood flowing to the penis, you don't have hydrostatic pressure, and therefore the penis cannot do the job that it needs to do. At the tip of the penis, there's the glans penis, and this is kind of the expanded distal end of the penis. And there may even be a foreskin if a male has not been circumcised. There will be a foreskin over the end of the penis. And you can see there's some parts that I did not highlight. There's parts that are affiliated or I guess near the male reproductive system, such as the ureters. Ureters will carry urine to the urinary bladder. You can see the urinary bladder is right there. So coming out of the urinary bladder, the male reproductive system and the urinary system share that common tube called the urethra. And you can see the large intestines in the posterior region, but it's not really associated with the male reproductive system. So that's a little bit of an overview. You know, if you want to know if you really understand the anatomy of the male reproductive system, you should get a blank piece of paper out and sketch it. So this is probably the best view to sketch most of the major parts. So I encourage you to stop the video and get a piece of paper and sketch it and try to see if you can remember the parts. So just looking at a few other kinds of drawings, just to kind of highlight a few extra things. Here we have a frontal view, an anterior view of the male reproductive system. Just to orient you, the penis has been pulled forward or superior, and we're looking mainly at the layers of the scrotum in this particular diagram. A couple things that are kind of interesting here is that, you know, the scrotum has, of course, the testes, the epididymis, and parts of the vas deferens. But if you notice, it hangs outside of the main body cavity. So the cord, the spermatic cord here, which is going to carry blood vessels, the vas deferens, nerves to the scrotum, it actually has to penetrate through the external oblique muscle. So there has to be a hole in that external oblique, and we see that at the superficial inguinal ring, there is a penetration through the exterior, the oblique muscle. So that wall of that muscle has to be penetrated so that the things can go through. Now, during development, the testes have to actually move through the superficial inguinal ring to get down and descend into the scrotum. Sometimes males' testes don't descend through the external oblique muscle. If we look at a few of the features, we can see that there is muscle tissue that surrounds the spermatic cord and the testes. That cremaster muscle are basically skeletal muscle tissue which raises the scrotum. So if a male jumps into ice cold water, the scrotum will rise because the skeletal muscles will actually contract. And that's kind of like a sperm survival mechanism. So the scrotum does hang away from the body to keep the temperature correct for sperm maturation. But if the sperm cells get too cold, they won't mature and develop in the correct way. So in cold circumstances, the muscle tissue will contract to bring the scrotum closer to the body, therefore keep the sperm cells warmer. If we get down to the layers of the scrotum, we can see there is scrotal skin. Okay, that's a stratified squamous epithelium, just like your regular skin. Underneath of that, there will be the dartos muscle. These dartos muscles are going to be smooth muscle tissue, and these also raise the scrotum under cold situations to regulate the temperature of the sperm cell development. So if we look at a picture here where we take and remove some of the skin around the spermatic cord, as you can see, is going to contain various kinds of nerves, veins, arteries, and the vas or ductus deferens. The vas deferens is going to carry sperm cells from the epididymis to the ejaculatory duct and then eventually to the urethra. So that's all you could really see in this one. So we do see the epididymis here. There is a cavity called the scrotal cavity that the testes sit in. And just like every other kind of part in the body, there are boundaries. So we have the visceral layer and we have a parietal layer. The scrotal cavity is in between the two layers. And this is just what a cadaver, kind of a a mid-sagittal section through a cadaver kind of looks like, so you can get a sense of generally what the penis looks like with all of its associated parts. You can see the spongy material there, or cavernous material that can fill with blood, so that you can have a penile erection. There are different parts of the urethra. The spongy urethra goes through at the end, and then we have the prostatic urethra goes through the prostate gland. So, and then we have the intermediate urethra is entering into the shaft of the penis. Okay, so you can see kind of like everything looks kind of tan, and just kind of get a sense of what that looks like in a cadaver. Well, the gonads or testes are the primary sex organs of the male, and they're going to produce sperm and hormones, specifically testosterone, and a couple other things that we'll talk about. So the testes are ovoid in structure. They're located in the scrotum. They contain about 250 lobules, and we'll see how those lobules are. These lobules are units of organization that are separated by connective tissue, and the job of the lobule is to hold 1 to 3 tightly coiled seminiferous tubules. The seminiferous tubules are the tubes that are going to create the sperm cells. So there is epithelium within that tube that gives rise to the sperm cells. In between the seminiferous tubules, there are interstitial cells, and these interstitial cells are going to be the cells that, under the right hormonal stimulation, will produce testosterone. There are channels that lead from the seminiferous tubules that carry sperm to the epididymis and to eventually the vas deferens. Something I find of interest is that each testis has, in my calculations, I've never seen it written out, but when I calculated it, they have about 2,133 feet of seminiferous tubules. That's tremendous. So, you know, a male, when he produces sperm cells, or when he ejaculates sperm cells in semen, there's hundreds and hundreds of millions of those sperm cells there. So you have to have a lot of these seminiferous tubules in order to produce enough sperm cells, and it takes 2,100 feet of seminiferous tubule to produce the level of sperm cells that have to be ejaculated. All right, so what you're seeing here are two different drawings. One of them is kind of a superficial drawing, so showing you the seminiferous tubules organized inside of those lobules. So there's a lobule there. You can see how they're kind of sectioned off right there. And the seminiferous tubules are these tubes that are coiled back and forth right there. So these seminiferous tubules, you can see they will all send sperm cells to this little network of channels. We'll look at the name of it in just a second. And then those will take the sperm cells to the epididymis. Eventually the epididymis will take sperm cells to the vas deferens. In the second drawing over here on the right hand side, we have a drawing of the seminiferous tubule cut in cross section. So you can see the interstitial cells lie between the seminiferous tubules. And then we can see the epithelium on the surface of the seminiferous tubule. So that's the surface, and then this is the lumen down here. This is the lumen down here. So at the surface of the seminiferous tubule, making up the wall of the seminiferous tubule, you have a special epithelium that's going to undergo spermatogenesis to produce the sperm cells. And then you'll see there's rows and rows and rows of cells as they go through meiosis or spermatogenesis. They are going to eventually produce sperm cells that will be in the lumen and can move toward the epididymis. All right, so this is if you look down here, you can see how this is planed through here. This is kind of a transverse plane through the scrotum and the testes, so we can get a kind of a different view of it. Again, you've already seen the cremaster muscle, and you've seen the I'm just looking through. So you've seen the scrotum and the dartos muscle. So we had multiple layers of muscle, again, to pull the scrotum closer to the body during cold situations. So something that we've already talked about, we already looked at the lobule. Okay, so the lobule is this little section right here, and the seminiferous tubules. The seminiferous tubules are those coiled tubes. Now they all lead eventually to the epididymis. So that's where everything's heading towards. So if you take a look over here at the ready testes, you have these tubes that all connect eventually to this particular section, and it moves it towards the epididymis. Now the epididymis is also a series of coils that are going to store the sperm cells and help them to mature. So just looking at it at a different view, this particular view is a sagittal plane through the testes. So you can see it's a sagittal plane through it, just to give you a sense of the lobule and the seminiferous tubules leading to the ready testes, which is the tubes leading to the epididymis, and then the epididymis will lead to the vas deferens. Okay, so sperm cells are made, they move to the epididymis, and eventually move to the vas deferens, and then they'll move out of the testes and move toward the ejaculatory duct. There are lots of blood vessels and nerves that feed this area, and that comes through the spermatic cord. And this is just what it looks like from a cadaver. So this is more of a superficial look at it. This is more of a sagittal look at it. Okay, so there's a lot of organs that are accessory organs to the testes, which is the primary organ. The epididymis is a tightly coiled tube lying adjacent to the testis and leading from the testis to the vas deferens. And it's going to be the site of sperm maturation. So sperm cells are made in the testes, but they're matured in the epididymis. Each epididymis is 23 feet long, so a male has 46 feet on average of epididymis. Okay, why so long? Because there's a lot of sperm cells to store, and of course maturation takes place there. So our next part is the ductus or vas deferens. The ductus deferens is a muscular tube. It's about 45 centimeters long, so it's almost half a meter stick long, and its length is pretty significant because it has to go back up into the pelvic cavity and loop around and bring sperm cells down to the urethra. It leads from the epididymis up into the body cavity to the ejaculatory duct, where it unites and empties into the urethra. And I'm pretty sure you've heard of a vasectomy before. A vasectomy is the cutting of the vas deferens. And there's different ways you can do it. You can cut it and stitch it. You can cut it and cauterize it with a hot iron. So there's different ways you can seal. But the reason we do a vasectomy is to prevent the sperm cells from being ejaculated out. So it's a form of permanent birth control. Now you have to be careful. After you have a vasectomy, there are still sperm cells that are in the vas deferens, and they can last there a pretty long time. So after having a vasectomy, sexual intercourse, there's a certain regimen of time that you have to abstain from unprotected sexual intercourse to make sure you clear the vas deferens of all the sperm cells so that an unwanted pregnancy does not occur. So the seminal vesicle is another accessory organ. It's a sac-like structure attached to the vas deferens near the base of the urinary bladder. And this seminal vesicle, also known as the seminal gland, it secretes an alkaline fluid containing several different things. One of the things it contains is fructose. Fructose is used to nourish the sperm cells so they can make ATP and continue beating their flagellum. And it also produces prostaglandins. The prostaglandins are thought to cause muscular contractions in the female reproductive tract to help basically draw in sperm cells closer and closer to the cervix so they can come up into the uterus and move toward the fallopian tubes. So it also contains clotting proteins. to help the semen clot in the vagina. So after a male ejaculates in the vagina, you don't want all the semen to run out. These clotting proteins will help the semen to clot together to stay within the vaginal canal. That clot won't stay there forever. It will be digested by other digestive proteins released by other glands. So the prostate gland is our next accessory organ. It's the size of a golf ball. Males only have one of them, and it does surround the urethra coming out of the urinary bladder. It's shaped like a donut, and so that the urethra can go through it. The prostate gland secretes a fluid of citric acid. The citric acid is used by the sperm cells to make ATP. It also contains proteolytic enzymes, the secretion coming out of the prostate gland that it adds to the sperm cells. These proteolytic enzymes help to digest mucus and also clotted semen. So these digestive enzymes don't work instantaneously, but they do work after a period of time once the semen is in the vaginal canal. So seminal plasmin is a protein that has antibiotic properties, and it's thought to maybe kill bacteria in the urethra, thus reducing urinary tract infections in males. I guess you remember from the urinary system that the male has a longer urethra, females have shorter urethras, and therefore females are more susceptible to urinary tract infections because of the length difference in the urethra. So here is just a view, this is a posterior view showing you the prostate gland. So let me get my highlighter function here so we can look and see the prostate gland, which would be the structure here. Again, it's kind of like a donut with the urethra going through it. And then we have the vas deferens or ductus deferens coming down. So sperm cells are coming down. They'll meet up in the ejaculatory duct. At the ejaculatory duct, there are certain secretions that are going to come into the sperm cells to make semen, and those are going to come from the seminal gland or seminal vesicle and from the prostate gland. So males, oftentimes the prostate gland is the Achilles heel of a male. It's very easy to get prostate cancer. It is it is a tissue that's epithelial in origin, and lots of our cancers are caused by are caused in epithelial tissues. So here's the prostate gland. Here's the urinary bladder, just to orient you. Here's the urethra coming through. Here's the rectum. So you can't go through the front side to test the sponginess of the prostate gland. A prostate gland is hard or is considered to be something diseased. So the prostatic massage or the digital exam is basically where the healthcare practitioner takes a finger, a digit, that's why it's called a digital exam, and they put it into the rectum and press on the wall of the rectum to feel the sponginess or how the tissue feels in the prostate gland. If the prostate gland doesn't feel well, and the PSA antigen test, which is a blood test for prostate cancer, if all these things come back negative or come back positive, that there's something might be wrong, they may have to insert a needle through here. So they'll insert a needle in and do a biopsy of the prostate gland. So the bulbourethral glands are the size of garden peas. If I just go back one slide here, here's your bulbourethral glands in this posterior view down here. So they're not real large, they're the size of a garden pea, and they're located inferior to the prostate, and their job is to secrete mucus, and this mucus will help to lubricate the tip of the penis also lubricate the urethra during sexual arousal. So this helps to reduce friction during sexual intercourse. It also secretes an alkaline fluid to help neutralize acids that are in the urethra and maybe to a smaller degree in the vaginal canal as well. I think I might have missed this, so I want to go back and make sure I emphasize this. Many of these fluids that come out of the prostate and seminal vesicle are going to be alkaline, and that is to neutralize the acidity of the vaginal canal. That acid will kill sperm cells, so we have to have a way to neutralize it, and we do it through the fluids that are added to the sperm cells. Now, semen is a combination of sperm cells and all the secretions of the seminal vesicles, the prostate gland, and the bulbourethral gland. All of that makes up what we call semen. Within semen, there may be somewhere in the order of 50 to 150 million sperm cells per milliliter of the ejaculate. pH is somewhat basic, so it ranges anywhere from 2.7 to 7.7, and the average volume per ejaculate is going to be somewhere in the order of 2.5 to 5 milliliters. Could be more, could be less. So let me talk to you now about the male external reproductive organs. So we have the scrotum. The scrotum is that pouch of skin which covers the testes. We also have the penis. The penis is that cylindrical organ that's made up of specialized erectile tissues. We talked about the corpora cavernosa and the corpus sponginosum just a second ago, but we're going to talk about it a little bit more here. Those are erectile tissues. They have the ability to engorge themselves with blood, and that basically forms a hydrostatic pressure that causes an erection to occur. The penis is designed to convey both urine and semen to the outside of the body. The corpus sponginosum enlarges at its distal end, at the very end of the penis, to form the glans penis. So here you can see the glans penis right here as it's basically enlarging. There's some ideas on why the penis is shaped like that. So if you looked at kind of a diagram of the penis, you can see that it has an enlarged end. It's thought to be like a plunger of sorts. So if males are competing over females for fertilization of her eggs, if a male leaves semen in the vaginal canal and then another male mates with a female, this particular structure right here may pull sperm cells out during sexual intercourse. So that's kind of through a competition. Sexual competition for depositing sperm cells and being the last to deposit sperm cells and removing all your competitor sperm cells during sexual intercourse. So this is kind of an anterior lateral view. You can see the corpus sponginosum. So it's this erectile tissue here all the way to the glans penis, and the corpora cavernosa is this erectile tissue here, making up kind of the main cylindrical portion. Of course, there's a root back here, so the penis is rooted in into the body cavity. This is a frontal section through. This is a frontal section through the tissues, so you can see the erectile tissues in this particular view, and the corpus sponginosum and the corpora cavernosa. You can also see how the glans penis is kind of enlarged at the end. So, and then you can see that in some males there will be a foreskin. Okay, that foreskin will be removed in a circumcision. So some males have foreskin, some males do not. It says in many textbooks, or indicates in many things that I've read, that if you remove the foreskin, typically it's done right after birth, that it reduces penile cancer and the acquisition of urinary tract infections, and it reduces the incidence of the spread of HIV infection. So it's still controversial as to should you remove it or not, because any surgery doing a baby is surgery on a baby, and it can have problems and things that go wrong. So if you notice here, we're taking a transverse plane through that. This is a transverse section through the penis, just showing you again there are arteries that feed that erectile tissue. Look at all the arteries that are coming through, and here's the corpora cavernosa. So this would be the corpora cavernosa, and then the corpus sponginosum. And that's the urethra going through. The spongy urethra goes through the corpus sponginosum. I always like to show what it looks like in a cadaver. You know, the artist renditions are always so beautiful, but that sometimes they look a little different than what the cadaver looks like. So you can here see the sponginosum, corpora sponginosum, and the corpora cavernosa of the penis. A little overkill on diagrams. I apologize for that. But again, one frontal plane just showing you the urethra. This is where sperm cells have to come out of. But you can also see the corpora sponginosum and the corpora cavernosa. So sperm cells are going to be our next topic, and we'll talk about how sperm cells are formed. So a sperm cell has a head containing a haploid nucleus. A haploid nucleus means there's only 23 chromosomes in that particular nucleus. Most of your, all the rest of your body cells have 46 chromosomes, which make you a diploid organism. Haploid is the name we use for a single set of chromosomes. Diploid is the name we use for having two sets of chromosomes. You know, two sets of chromosomes, one set was given to you from your mother, one was given to you from your father. So the sperm cell is a very specialized cell. It has that haploid nucleus. It contains a mid piece or a middle piece, and that contains mitochondria, so they can generate ATP. And then it has a tail that is what we call a flagellum, and it has the ability to move this flagellum so that it can thrust forward the sperm cells. At the tip of the head of the sperm cell, there is a part called the acrosome. And this is a bag of digestive enzymes that will help erode the tissues that surround a female's egg cell. And when we get into the development lecture, we'll look at how that process actually occurs. So here is the acrosome, that bag of digestive enzymes. There's the nucleus with the 23 chromosomes. Here's the middle piece, mitochondria not being shown. And here is the flagellum. The tail is made up of a flagellum. All right, so spermatogenesis is a pretty complex phenomenon. I'll show you some pictures and kind of go over it with you. You may have to stop and relook at the video once or twice just to get a sense of kind of what's occurring. In the male embryo, spermatogenic cells are undifferentiated, and they're called spermatogonia. So even in the male embryo and fetus, the spermatogonia are already developed and ready to go. Each of these spermatogonia contain 46 chromosomes, so they are diploid. Now, the thing about fertilization is when a sperm cell and an egg cell come together to make a human, to make a zygote, you want each of those to have 23 chromosomes, because the 23 chromosomes added together make 46 chromosomes, and that's how many chromosomes need to be in a human being. So if that zygote does not have 46 chromosomes, it won't develop correctly. It has to have 46 chromosomes. So we have to have a way to take cells, stem cells that have 46 chromosomes, and make them into cells that have 23 chromosomes. And spermatogenesis or meiosis is going to be the process that does that. So during spermatogenesis, spermatogonia will enlarge and they will become primary spermatocytes. Primary spermatocytes will undergo division by this process called meiosis, and they're going to form eventually 4 haploid secondary spermatocytes with 23, they will form 2 haploid, one primary spermatocyte will form 2 haploid secondary spermatocytes with 23 chromosomes. So it kind of looks like this if it just kind of diagrammed it out. So the spermatogonia will become primary spermatocytes. These will divide into secondary spermatocytes. And if let me back this up just a little bit and let me ink this off a little bit. So the spermatogonia will produce primary spermatocytes. They will undergo division to produce secondary spermatocytes, which will undergo a second division. So this was division 1, this is division 2, to produce your sperm cells at the very end. So the spermatogonia will eventually divide to form 4 haploid spermatids or spermatocytes. spermatids. Okay, now we have to do this because we start with 46 chromosomes, and this reduction division that happens will end with each sperm cell having 23 chromosomes. So secondary spermatocytes will divide again, so that'll be meiosis 2. Meiosis 1 was the primary spermatocytes undergoing division to form the secondary spermatocytes. Meiosis 2 takes the secondary spermatocytes, divides them to form spermatids, which will eventually mature into sperm cells, functional sperm cells. Now this is a cut through the seminiferous tubule, so you can see the interstitial cells. Those are going to be cells that under right hormonal stimulation will produce testosterone. You can see in the wall of the seminiferous tubule there are spermatogonia. So all along this wall there are spermatogonia that are stem cells that are going to undergo division and mature into eventually these spermatids. So there are nurse cells that surround and aid all of the maturation process of these sperm cells. If I just give you a little bit deeper view of that, so you can see the interstitial cells there that are going to produce the testosterone under right hormonal stimulation, but at the very basal part we have the spermatogonia. Those spermatogonia will turn into primary spermatocytes, and primary spermatocytes will undergo division to create secondary spermatocytes, and secondary spermatocytes will undergo division to produce spermatids. And then the spermatids will basically go to the lumen of the seminiferous tubule and head towards the epididymis. Now it's important that you see this cell right here. There is a cell that, let's see if I can highlight it. Let me move myself and highlight it. So there's a cell here that you can see, if I get my highlighter function right. So there's a cell here that you can see, and this is called a nurse cell. It's got some different names in books, but nurse cell is the way I like to think of it. And so it's this whole cell right here, and it kind of wraps around these sperm cells as they're developing. And so that's the whole nurse cell that you can see there. One of its jobs is to attach to other nurse cells through tight junctions and create what we call the blood-testes barrier. Okay, so you're familiar with probably the blood-brain barrier that protected the brain from materials in the bloodstream. The blood-testes barrier protects your body from being exposed to new kinds of proteins that are developing on the surface of these spermatids and sperm cells. So there are unique proteins there that the body's not familiar with. If these sperm cells get into circulation, you will produce antibodies against the sperm cells. Okay, so this blood-testes barrier protects the body from being exposed to those unique proteins. I kind of like this diagram because it just kind of shows the nurse cell wrapping all around, sustaining, helping to nourish, helping to allow this process of spermatogenesis to occur. It's a really complex cell. Okay, so I really appreciate that cell. It's an important cell to the spermatogenesis. So here is spermatogenesis once again in the graphic. You know, you may need to stop the video and just kind of look at the graphic for a second, get a sense of it. But primary spermatocytes undergo meiosis 1, the first meiotic division, and they're going to divide themselves into secondary spermatocytes. So that's a secondary spermatocyte there and there. Each of those is going to have 23 chromosomes. They're going to undergo meiosis 2. So the secondary spermatocytes undergo meiosis 2 to produce spermatids. Each of them produce 2 spermatids each. Okay, so at the end of this spermatogenesis, at the end of meiosis, you're going to get in males from one cell, you get 4 cells. You get a cell that had 46 chromosomes and cells at the very end that only have 23 chromosomes. Remember, we have to have 23 chromosomes because that's the number that when it adds to the 23 chromosomes of egg cells creates a perfect human of 46 chromosomes. This is just a microscopic view of the seminiferous tubule. So there are tremendous numbers of sperm cells that are being produced by these. You can kind of see what spermatogonia look like. They're going to be more on the outer edge of the seminiferous tubule. There's those interstitial cells which will produce the testosterone under the right hormonal stimulation. All right, so this is the sperm cell, and I just kind of highlighted here the acrosome is that little bag of digestive enzymes that help with penetrating the egg cell. Here's the neck. Here is the middle piece. It contains the mitochondria for producing ATP, and then the end piece or tail piece is going to be used for the movement. That is the flagellum that aids in movement. Now, during sexual arousal, the parasympathetic nervous system is going to trigger an increase in blood flow to the erectile tissues of the penis. That's going to produce the erection. The culmination of sexual stimulation will lead to an orgasm. During an orgasm, you'll have the emission or movement of sperm cells and accessory gland secretions into the urethra, and eventually that will force the semen outside through ejaculation. After ejaculation, the sympathetic nervous system now takes over, and it constricts the arteries that are going to the erectile tissues of the penis, and that will return the penis to its unerected, flaccid state. So if you've ever seen the erectile dysfunction medications, those are working because they dilate blood vessels, which allow for the flow of blood to the erectile tissues. Now, the hormonal control of the male reproductive system is pretty complex. There will be hormones secreted by the hypothalamus that will go to the anterior pituitary gland. The anterior pituitary gland will secrete hormones that will go to the testes, and those will basically control male secondary sexual characteristic development and male reproduction. So at the time of puberty, the hypothalamus releases gonadotropin-releasing hormone. That triggers the release by the anterior pituitary of luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone is going to promote the development of the interstitial cells, which will help them to begin to secrete testosterone. Follicle-stimulating hormone plus testosterone will stimulate spermatogenesis to occur. So male sex hormones that will be produced by those interstitial cells are the androgens. Okay, testosterone is the most abundant, and it's secreted. It's oddly enough secreted in a fetus, so you can get masculinization of the fetus, so that it develops the right parts, and then it's halted, and then not until puberty does the release of testosterone begin again. Once testosterone begins to be released, it's continuously secreted throughout a male's life. Again, you may want to stop the video here and just kind of look at this diagram for a minute. But we'll start up here at the hypothalamus. The hypothalamus is going to release gonadotropin-releasing hormone. That goes down through that portal vein system, the hypophyseal portal vein system, to the anterior pituitary. At the anterior pituitary, follicle-stimulating hormone is released and luteinizing hormone is released. Luteinizing hormone targets the interstitial cells of the that are in between the seminiferous tubules, so it develops them and helps them to release testosterone. So the follicle-stimulating hormone plus testosterone will begin to stimulate the spermatogenesis to occur. So those nerve cells are kind of interesting. They are going to release inhibin, and that will be a negative feedback loop to kind of control the release of gonadotropin-releasing hormone. High levels of testosterone will also negatively feed back in to inhibit the release of gonadotropin-releasing hormone. Now, this testosterone is the real deal. Okay, so it's going to, in addition to follicle-stimulating hormone, it's going to help spermatogenesis, but it's going to have many effects on males, and we'll look at those effects right here. So it stimulates the development of male reproductive organs, causes testes to descend. Testosterone is also responsible for male secondary sexual characteristics like deep voice, body hair, thickening of skin, enlargement of muscle tissue, development of sexual function. It stimulates protein synthesis, and so lots of things are occurring because of the action of testosterone. So we do have a negative feedback system. It involves testosterone and inhibin. Both of those will go back and block the release of gonadotropin-releasing hormone. Okay, so we're going to stop there, and next lecture we'll pick up with the female reproductive system, which is the harder of the two. So until next time, I will see you later.